is from Dr Louis Tremblay, who is an ecotoxicologist and colleague of mine at the Cawthron Institute in Nelson. Louis is a good runner and scientist, and he co-leads the MBIE-funded Emerging Organic Contaminants project. And this is going to be the topic of his presentation today. So welcome, Louis. We look forward to hearing what you got to say. Feel free to share your screen.
Let's try this.
Perfect, mate.
I'll go. Perfect. Kia ora tātou, and welcome everyone. Thanks very much, Tim and Catherine, for inviting me to be part of that webinar. I've just had a 2-day long webinar on as part of a conference that was based in Melbourne. So I don't know about you guys, but I'm starting to be a bit suffering from Zoomitis, but I'll try to do my best to, like Tim mentioned, so I lead this MB program on emerging contaminants, and you will see that there are quite a bit of relevance for food safety, because some of those chemicals can, partition in some food where they can pose a risk. And I should mention before I start that I'm part of a larger group as well. So I have colleagues across those different organizations, and I put their logo down on that slide. So just a quick overview of what I'm going to talk about. I'll talk a little bit about what it is, what's an emerging contaminant, and what are the challenges when we're trying to assess their risk. What are their risk and knowledge gaps, the science that we're using, and I'll talk about the national strategy to manage emerging contaminants that we recently launched as part of this program, and hopefully talk a little bit about what's coming, what's over the horizon in terms of solution. And I see that Jan and Phil are talking about plastics, so there's going to be some relevance as well to plastics, because I'm part of the ESR-led AIM2 program. So what we can consider an emerging contaminant is any contaminants that enters the environment that is not traditionally monitored or regulated. So as you can see, there's a whole lot of chemicals that are being released into the environment, from the personal care products, cleaning products, and in here you have the firefighting foams, and for some of you may be familiar with PFAS, those very persistent chemicals. And then the problem or the challenge from an ecotoxicology perspective is that there are lots of them, and there's really not a whole lot of information about their fate into the environment and the effects that they have on exposed biota. So here's a list of what could be considered emerging contaminants. Obviously, you have the pharmaceuticals, industrial chemicals like PFAS, agrochemicals. We're using a lot of pesticides. And then the other one that I was hoping that Jan would talk a little bit more about is that, one of the problems with plastics and microplastics, or that could be the elephant in the room, is that they all contain chemical additives. And here there's a little illustration of why we're adding chemicals in plastics. It's to give them those different characteristics, the strength that they have, or the flexibility. If they're to be used in an outdoor situation, they need to have UV blockers. They have colorant, stabilizers, and so forth. And sometimes they can be up to 40 and 50% of the plastic itself can be chemical additives. So we published a a review on the risk of those on the risk and knowledge gaps about the plastics in the chemicals in plastics. And like I mentioned before, I think this is really well articulated by Andrew Johnson and his colleagues in the UK about what is the main challenge of dealing with emerging contaminants. It's about the sheer volume of chemicals on the market and the ones that are being continuously introduced. And it rapidly outpaces our ability and capability to assess their risk. And so we have to take that into account of how we can address this issue. So I just want to quickly show this, the structure of our program. So in the dark blue in the middle here, you have the actual science. The science, because it is a complicated topic, so therefore it requires a multidisciplinary approach. So Grant Northcutt and his group are looking at the chemistry. Here at Cawthron, we're doing the ecotoxicology, but we also have a strong component on vision Mataranga. We have also colleagues in Australia. Our colleagues at ESR at Massey Universities are focused on antimicrobial resistance and the role between emerging contaminants and the modulations of AMR. And we have also a strong community engagement because we're conscious that we are all as individuals contributing to the problem as well, so we should be part of the solution. Another feature that I'm going to talk about is that we've developed a national strategy to manage emerging contaminants. And then we have 2 study sites that I'll talk a little bit more about. And then a key feature is probably that national advisory panel. And that's quite important because that panel has membership from across key regional councils. And those people are also experts into particular environmental compartments, that is surface water, groundwater, soil, health, and coastal. But also we have representation across the central government, MFE, EPA, and Jan here has just replaced recently Andrew Pearson, that's moved to the private, he's moved to the private sector. And then we have also a Maori representative, and Hardeep, he's our industry representative from Sanford. So the role of that group is really to monitor the progress of our research, make sure that we remain aligned with you know, the key concerns that people have, and then also that we have a level of agility to address new issues as they emerge. So those are the 2 case studies that I mentioned before. So we have the Southland, and then we have the full catchment on in the Auckland region, in the Auckland city actually. And those sites were selected for a range of reasons. So up in the full, obviously it's a highly populated, high density area with a range of sources. And then the catchment in Southland is slightly different. It ends up here at the New River Estuary. So it has a little bit of urban input as well, but a range of agricultural input and light industry. And the red dots here are all the areas where we sampled the environment using the passive sampling devices that I'm going to talk about to try to give us an idea of what are the chemicals that are there, which ones are the ones that are persistent, with the aim to come up with a long short list of those more relevant or persistent chemicals that we should follow up in terms of further characterization of their risks. So those chemicals are, you know, usually found in large numbers. So you can expect in an environmental sample to have like hundreds, maybe in the thousands of chemicals, but they're found at very low concentrations and continuously released. So what those devices are doing is that they are capturing those chemicals as they come into the water based on their chemical characteristics. So we're using different ones so that are capturing different families, if you want, or range of chemicals. And we even use plastics. Like as Jan mentioned before, one of the key things with plastics is that they can actually absorb some of the chemicals in the ambient environment. And then again, those chemicals have a certain chemical characteristics. So what do we do? So we deploy those samples, those passive samplers in the river, in the catchment, and then this is what it looks like. So you have the different devices again that are capturing those different range of chemicals, and then here that gives you an idea of the size. So here's my colleague Grant Northcutt, and then this is a colleague from Environment Southland that's helping us to deploy them down in Southland. So what do we do then with an environmental sample that has a lot of chemicals? So we can't just go straight to chemical analysis. So what we're doing, we're using this effects-directed analysis approach, where we first extract the environmental samples, and then we test them with a biotest, with bioassays. And then the ones that have the highest activity are further fractionated and again tested again. And the idea is to start with a very complex mixtures of chemical and slowly bringing them down to a number where we can actually use chemical analysis to identify the individual or families of chemicals that are responsible for causing the biological activity or the harm. So those are the bioassays that are used. So this is with our colleagues from Griffith University in Queensland. So here you have the range of assays, but those assays are selected for different endpoints. So this one is giving an idea of toxicity to bacteria. This one is providing toxicity to plant, and then different biological activities here. So this is an indication of dioxin-like compound activity, endocrine disruption, and then here genotoxicity. So any contaminants that can interfere with the DNA. So what we do, we set our passive samplers in the environment. We bring them back after 3 weeks to the lab. We extract them, and those extracts are split, and then some of them are sent to Griffith University, and they're tested with those different that range of bioassays, and they're ranked according to the level of activity, so high, medium, or low. And then that gives us the information that we need to rank the samples in relation to their biological activity that are then selected for further fractionation. So here you have the type of device that was used, the site where it was, and then its ranking. And then those that have a yes here for fractionation have been selected for further fractionation. So we're doing it. So this one is for the passive samplers from Southland. We have the same type of information for the sediment, because sediment are natural passive samplers themselves, because most of the hydrophobic compounds will partition in the sediment. And then we have it for Southland and for the FO. So this is where we're at now. We've had quite a few delays, and because of lockdowns, when it was not here, it was in Queensland, but we've made significant progress in the last few months. So the idea is then, once we're at a level where those samples are ready to be chemically analyzed, so we're going to use a range of targeted analysis. So for those analysis, we use a reference chemical. So we specifically measure those ones, but a lot of those chemicals emerging contaminants are not known. So we're also using what we call a non-targeted analysis. So that provides a look into some information about what is likely to be there. So then we can go back and using targeted analysis. It gives us also the ability to not only look at the parent compounds, but also the persistent metabolites. From the toxicity testing perspective, so this is what we can use to sort of rank further those chemicals. So what we've done here, we know that those pharmaceuticals and personal care product chemicals are found in our sewage water. So what we do, we go back to the original compound and then use different bioassays. So we use here a copy pod toxic assay, and then 2 muscle embryo larval development assays. And that gives us an indication of which are the ones that are more toxic. So here, triclosan, which is, you probably heard of it, is actually very toxic compared to metropolol and some of the other ones. So that gives us, again, an opportunity to, an option to sort of reduce a large number of chemicals, so we can focus on those ones that are more posing the highest risk. Another important component of some of the emerging contaminants is that there's little information in terms of their mode of toxicity. So we're using anomics approach, and that's a very nice little graphic here showing the cascade of event in a cell. So from the transcriptomic, or the transcription of the messenger RNA, and then translation into the different proteins and enzymes. So we're using some of those steps to try to identify what are the physiological processes that are targeted by those chemicals for which we have less information. And here, this is a little workhorse, if you want. It's a little New Zealand copy pod. And the reason we're using it is that it has a very short life cycle. So within a month, you're going to have a full life cycles from baby to reproduction. So here you can expose the same organisms over multiple generations. And the reason is that a lot of those chemicals, a lot of the EOCs are not very acutely toxic, but their effects are likely to be demonstrated over a longer period of time, over chronic exposure. So that gives us a really good model to sort of look at what are the multi-generational impact of those chemicals. So here we use, in an initial experiment, we use benzophenone, which is both in microplastics as a UV blocker, diclofenac, which is a drug, and then copper. And then what we do at the end of the 3 generations, we isolate the DNA, and then we can look at the transcript of, or the profile of expression of different genes to try to identify the genes that are, or the main physiological processes that are affected, being reproduction or metabolomic processes that can then be used to develop what we call biomarkers, because then we can use those biomarkers to go back into the field to see whether the biota that are exposed, whether they are exposed to those particular chemicals. So this is how this information on transcriptomic can be used. Here you have the different pathways, and then here you have the different enzymes and proteins. And then here that gives you the information about whether those genes are upregulated if they're red, or downregulated if they're blue, in relation to the control. So that gives us a really precise information in terms of what is happening to this organism that is exposed continuously to that chemical. And then some of those enzymes that are the most affected can then be used or targeted for biomarkers development. Another component of the research is around the role of EOCs in antimicrobial resistance. And this is our colleagues from ESR and Massey University that are leading that objective. And here, the question that we're asking is, can EOCs or some EOCs promote AMR in the environment? And if they can, so which ones are the ones that we should be concerned about? So some of the outcomes that we're expecting is to elucidate the mechanisms behind those resistance, the development of resistance, and then, start engaging as well in terms of, antibiotic molecule stewardship, identify hot spots, and then the selection pressure as well. And then to a certain point, to highlight the priority chemicals, how can we better manage them? Is it the question of ban, or is it a better control of the waste, etc.? And then Izzy Alderton just published a very nice review recently on the role of EOCs on AMR. We also have, we're also working with social scientists and Vision Maturanga Māori scientists, because this concept of wicked problem applies really well to emerging contaminants. So those are complex, symptomatic of other issues. They're often competing worldviews, and then they also, those seeking to solve the problem are also causing it. So we're all using chemicals, so therefore we're part of the problem. So, but when you have a range of people with different perspectives, it can be very challenging when it comes time to manage particular chemicals. So we take a Maori approach to this, is that we're trying to have an ecocentric model where we look more into our relationships as humans with our surrounding environment. So really going from the more Western approach of how we're dealing with the environment to a more eco, where humans are part of the wider ecosystem. So it's really a systemic approach that we're using, which is based on respect, responsibility, reciprocity, relationships, and also our role as kaitiakitanga. And then one of the great outputs from this program was the release of this Aotearoa New Zealand strategy for emerging contaminants, and that was launched a couple of months ago by Graham Civic Jones from Manaki Whenua and my colleague James Ataria. And so this strategy is providing the high level inspirational goal that we want to achieve, and we take that whakatoki from that translate into a sail the Leviathan while it is young. And then if you look from a historical perspective, the way we've introduced chemicals, if you look at DDT, for instance, or more recently PFAS, the original people had no idea what the problems the sort of problems that will end up with, accumulation in biota and so forth. So if we can be a little bit more proactive in the way that we use and introduce new chemicals. So in conclusion, I think we shouldn't forget the fact that chemicals have enabled major improvement in food production and keeping us safe and healthy. Unfortunately, there are some unintended consequences, and there's quite a few evidence of harms to our receiving ecosystems. So I have colleagues a few years ago that came up with that underpinning philosophy of do no harm, so ensure a sustainable, safe chemical environment for the future. And then what's the way forward? I guess it's quite important to realize that this is a global issue and that it does require coordination of capability and resources. And that was something that was discussed quite at length in the last 2 days at the conference that I was at. It is a wicked problem, so multidisciplinary approach is required. It requires also a cohesive approach and involvement from cross sectors. I'm just thinking about, the likes of maybe the initiative of MPI around ARIS, for instance. I think it's very important to work together. And then more and more, it's to take the One Health approach. We're working now increasingly with our colleagues from the health sector. Particularly at the moment, we're working with anesthetists, because they're concerned about some of the drugs that they're using and the potential harm to once it's released. And then the solutions must, take into account the balance between making sure we benefit from the chemicals while reducing the unintended impacts. Thank you very much for your attention.
Okay, thanks, Louis. We've gone a little bit over time, but you'll be able to take one question. This is by Barry Peake. By far, the highest proportion of your emerging contaminants are organic in nature, but do you also test for inorganic ones, such as trace metals? There's a little bit of subtext there around cadmium levels and water runoff from farms using fertilizers would be expected to be relatively high.
That's a good point, Barry. Thanks for that. We were very conscious when we put together the proposal that we needed to focus on one category of chemicals, and then one category being quite wide. So we're focusing on the organic chemicals, but we don't. You still have to recognize the fact that metals are still can be potentially emerging contaminants.
Okay, thanks very much, Louis. Just in the interest of time, we're going to keep moving. Really good presentation. Interested in your comment about the emerging risk identification system that the Food Safety Centre is establishing at the moment, and how contaminants that are observed or picked up through your program will feed into that. So really keen to connect the dots there.